Okay, guys. So, you're in cell biology. Just in case anyone didn't figure that out yet. Okay, so, business. No recitation next week. Because it's Rosh Hashanah and I'm going to visit my 97-year-old grandfather. So yay for you. No Monday, no Wednesday. But Friday. So I'm going to do some pre-recorded lectures online so you're not completely off. But, and then we'll have Friday to be a Q&A to kind of go over the stuff. So please watch whatever I post. I'll send out a reminder that you should watch stuff. And then we'll go over everything on Friday. Don't forget to bring Scantrons to recitation. Most people are remembering. Some people are forgetting. If you don't have your Scantrons for your quiz, you can't take the quiz. So you really want to do that. And then don't forget there's a quiz this weekend. Most people, no one's really forgetting. I've only encountered a few people who appear to be forgetting to take it. So that's good. Okay. So far we talked about what needs to happen to get a functional protein. Yes?
Sorry, I'm confused. We don't have class on Monday. We don't have lecture on Monday or Wednesday, but we have lecture on Friday and no recitation on Friday.
No recitation next week for anyone, because if I don't do it one day, then everyone's off schedule because it's by week. Yeah. Does anyone have any questions about anything I just said? It's in the syllabus. It's all in the syllabus. So what's really cool is I can tell who's not in class because I get these really strange questions that I spoke about in class and are in the syllabus and all that. But anyways, okay. So we spoke about what needs to happen or not happen to get a functional protein, and that what we were talking about is all those RNA editing, alternative splicing, RNA degradation, stability, all that. And then we talked a little bit about protein folding and how chaperones are important for that. We spoke a bit about ubiquitin because that kind of follows that when you have misfolded proteins, they're going to be ubiquitinated and degraded in the proteasome. But then, of course, I harped on the fact that it's not about misfolded proteins. It's just generally a lot of proteins need to be degraded to turn things off, and we'll talk about that later. And we ended with talking about microsomes. Does anyone remember what a microsome was? Piece of the ER. Great. Okay. Okay. So now we're really starting to get into talking about getting proteins to where they need to go. So we're starting with getting proteins to the ER. And so I like how in the textbook they have this kind of scheme that shows the flow of proteins. Right, so cytosol, so proteins can go from cytosol to the nucleus, cytosol to mitochondria, cytosol to plastids or peroxisomes. So those are proteins that are translated on free ribosomes in the cytosol and imported into those places. And then, as you notice, cytosol to ER, that's kind of like a layover that a lot of proteins need to take in order to go, and then to the Golgi to go to the endosome, lysosome, secretory vesicles, or cell exterior. Okay, so for those of you who are, because I've definitely had a few people who are like, oh, I'm overwhelmed, there's a lot of information and I'm having a hard time organizing it. So I'm trying to start each lecture with orienting you and trying to help put things in perspective. But please, if you're still feeling like things are just random information that doesn't attach at all, talk to me because I want that to not be how we start. Okay, so now we're talking about the ER and those who've had recitation. We talked in gory detail about import or export, actually. The author really harps on it being export from the cytosol to the ER. Really, you can think of it as just delivery of a protein to the ER. Okay, so proteins can be delivered via a post-translational or co-translational mechanism. This is all review, right? Because you guys read it for recitation. A third of you have already drawn it out in recitation. So this is pretty much on Monday what you guys did is you learned about co-translational translocation. So translocation of proteins into the ER at the same time that they're being translated. So the ribosome is actually dragged over to the ER with the mRNA, and the polypeptide is inserted into a translocon, and it's translated right into the ER lumen. And then post-translational translocation. Post-translational, obviously exactly what it says, post after translation. So the full protein is translated on free ribosomes in the ER and then imported into the ER post-translationally, right? Any questions about this major concept? It's a significant concept. So if anyone's sitting there going, I have no idea what she's saying, raise your hand, please. Don't let me go on. Okay, good. Everyone's going to get an A on the test. Oh, no, she mentioned test. There's going to be a test one day. Okay. So forget for a minute about post-translational import because it's not very exciting to me. Co-translational import is a lot more interesting to me. So in co-translational import, nascent, which is another way of saying new, nascent polypeptides and their associated ribosomes are targeted to the ER through recognition of a signal sequence. Blah, 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 blah. That basically means that a signal sequence is recognized on the peptide and the whole entire thing is dragged over to the ER. Okay? So this is what a signal sequence looks like. So these are your amino acids. So methionine would be your first. And then here it's showing some common, it's more of a consensus, not a consensus sequence, but it's more of like a pattern of amino acids than an exact, this amino acid means signal sequence, but don't worry about it. I will never ask you to draw me a signal sequence. And then it ends up being cleaved in the ER, so there's a cleavage site here. Okay, don't worry. Okay, so this is where we ended the other day. We were talking about isolating rough ER and how this is useful for studying import of proteins into the ER, right? Like little ERs that you can play with in the test tube, right? So when you disrupt your cell, you get microsomes, these little spheres of lipid that are coming from the ER membrane, and you can centrifuge them, and they will band in two different bands based on their density, right? And as you guys so geniusly told me on Monday, the rough microsomes are on the bottom, right, because they're heavier, and the smooth microsomes are on the top. What are the rough microsomes? Rough ER. Okay. So what's going to be useful for studying import of proteins into the ER? The rough ones, right? Okay. So this is how you isolate them. So why do you care, right? So, and this is just another figure about how it's done, because sometimes multiple figures help to explain things, right? So you disrupt, put in the tube that goes in the centrifuge. You actually have a gradient of sucrose. So basically think of putting it through something viscous, and you centrifuge, and then you get your smooth on top and your rough on the bottom, and you can pull them out. You just take a pipette and suck them right out, and put them in a tube, and you got your tube full of rough or smooth or whatever you want microsomes. And this technique is very common. Take microsomes out of it. People do this to isolate organelles, right? Because let's say you want to isolate mitochondria. Could this be a way to isolate mitochondria? Could this be a way to isolate nuclei? If they're not destroyed. So if you gently break open cells and you have intact nuclei and mitochondria and everything, and you put it on top of the sucrose and you centrifuge it, they're going to separate based on what? Density, right? And so nuclei are likely to have a certain density, and mitochondria are going to have a different density, and then you'll have microsomes in there too, probably, right? It's also a really interesting way to isolate protein complexes. I've done this in the past. If you want to say this protein interacts with this protein in this process, chances are they're going to interact together. So you take your cell lysate, you put it on a sucrose gradient, and you centrifuge it. If they're binding together, you're going to find them in the same band together, right? Because they should be associated, right? Okay. Anyways, that's enough about that. Okay, so we're talking about microsomes and why we're using them. So we're using them to study the ER function. So if you've got microsomes and they've got attached ribosomes and you treat them with detergent, what does detergent do to membrane? Breaks it down, right? So now you've popped open the microsome, right? If you add protease, what does it do to proteins? So basically, if you treat with detergent and you add protease, you're going to digest your secretory protein, which was inside the microsome. If you add protease alone, you won't have any digestion. So that's just saying that your secretory protein is inside your microsome, right? Makes sense because the secretory protein is going to be inside the ER, right? You've translated it into the ER, okay? So what's all this have to do with? So this kind of assay can be used to study the signal sequence hypothesis. So before we understood what a signal sequence was, people had a hypothesis about something called a signal sequence, right? So this is the sequence on the polypeptide that's going to target it to the ER, and then it's going to be cleaved off. So to test this, they did an experiment. They did in vitro translation. In vitro translation, that just means adding ribosomes, mRNA, tRNA, and amino acids, and you're going to get translation. I've done this. It's possible. It can really be done. It's a kit. Buy it from Promega. It's awesome. It's easy. Anyways, in vitro translation, and you get this peptide, right? If you do this in the presence of microsomes, you get a smaller sized polypeptide. So what accounts for the difference in the polypeptide length? Why is it smaller when you have microsomes and larger when you don't have microsomes? What are the microsomes doing? So we talked a little bit, yeah?
They're cleaving the signal sequence.
They're cleaving the signal sequence, right. Right. So we talked about signal sequence, and we talked about how there's a cleavage site and it's cleaved off in the ER. And we'll go more into detail about this. But exactly, so that means that the enzyme that cleaves the signal sequence lives inside the ER. And without these microsomes, it's not there. And so you'll just get, if you can still translate a protein, as long as you put ribosomes, RNA, tRNA, amino acids, you'll translate a protein, but it's going to contain the signal sequence, which won't be cleaved off. Yes?
Is it ever possible to export the protein into the ER without cleaving the signal sequence?
So is there any time that the signal sequence is not cleaved? I don't know. I guess it depends on if there's a cleavage site. So are you saying maybe that sequence might also serve as some other function?
Or I'm just saying, is the act of cleaving it what required for the import?
No, because it's after. So thank you for that question. So the question is cleavage required for import, right? So this experiment here is showing you that actually the cleavage is happening inside the ER. So it's being imported, and the enzyme lives inside the ER and cleaves it up. So it's already being imported regardless. And there are frequently signal sequences can act as start transfer sequences, which we'll talk about, but in membrane proteins, they'll become the transmembrane domain. Sometimes they're cleaved, sometimes they're not. So that would be an instance where it's not cleaved. But either way, the signal sequence is what's going to drag, as we'll learn about, it's bound by the SRP, and it drags the whole thing over to the ER. Okay, does that make it clear? Okay. Questions? Okay. Here's a practice problem for you. So everyone get ready to talk to your partner. You're studying IgG, an antibody secreted from B cells in the immune response. In vitro translation of IgG yields a protein that has a higher molecular weight than the protein that's secreted from a cell. Addition of microsomes to the in vitro translation experiment results in an IgG that has a lower molecular weight, identical to that secreted from B cells. Explain these results. So sit, read, think, chat. Does anyone feel this is an unfair question because they've never taken immunology? Okay, good. Just making sure you guys aren't getting swayed by my distraction of it being about antibody. Okay, chit chat for a minute or two, and then give me your answers. These are the kinds of questions that are on my exams. So.
Sorry, I'm just trying to figure this out. I think that's all it is. It means a musician that everybody's using. Wait, no. It's a longer batch. Yeah. That one side, you should say it's longer now. With it. So why would this, so would that be her alone?
Okay, guys. So I heard some things that sounded like people knew what they were talking about, and then I saw a lot of confused faces. So, how do you tackle this kind of question? Does anyone want to tell me anyone who feels confident about their answer, want to tell me how they started to think about this question? Okay.
So we just kind of thought about what we had just learned about the platelets that we
Right. So it's basically the easiest question ever if you followed what I was talking about. Who felt like they had no idea how to even tackle this problem? Oh, okay, great. So then if this appears on the exam, you'll all get it right? Okay, good. I'm seeing no's. I'm seeing yeses, I'm seeing no's. Shake your head yes if you're going to get it right on the exam. Shake your head no if you're not going to get it right. Okay. Okay, so for those who are shaking their head no's, first of all, thank you for your honesty, because it's honesty about what you don't understand that helps you to learn. When you lie to yourself, then you don't get the question right. Okay, so the best way to tackle this is to say to yourself, what the hell is she talking about? And then after that, say to yourself, okay, so she's talking about secreted. So that automatically should make you think circulatory pathway, something that had to go to the ER, right? And then she's talking about in vitro translation, so that's in vitro, outside of the cell, no ER is present. We're getting a high molecular weight protein, so larger protein. And then I say addition of microsomes. Okay, so then you say to yourself, microsomes, those are little ERs. Addition of microsomes results in IgG that has a lower molecular weight. So I'm telling you, if you add the ER in, you get a smaller protein. So then you have to ask yourself, well, what's in the ER that's making the protein smaller? And then you remember, well, to go to the ER, you have a signal sequence, and it's cleaved off in the ER. So So it's going to be smaller, right? And so what's secreted, so antibodies are secreted out of cells, right? So what's secreted from the cell is going to be the mature form of the antibody, not with a signal sequence, right? So that's why the protein has a higher molecular weight than the protein secreted from a cell when you don't have microsomes there. So if anyone has questions about this, please see me during office hours, make an appointment with me. I make myself very available. So if this shows up on an exam or a version of this, no one has an excuse, and I will individually go to those of you who get it wrong. Okay. Okay, so the SRP binds a signal sequence on the nascent polypeptide. It pauses translation and drags the whole complex to the ER. The whole complex. What do I mean by the whole complex? Ribosome. Ribosome, polypeptide, mRNA. The whole thing gets dragged to the ER. Does the whole thing get imported into the ER? No. Okay, just making sure. And so the way it looks is here's your polypeptide and has a signal sequence here, and your SRP binds to it and binds to that, and also the ribosome, and the whole thing is directed to the ER and where it binds to a SRP receptor. And this is another way of looking at it. So you've got your ribosome, your mRNA, your polypeptide coming out with your signal sequence. It's bound by your SRP, which drags it over. Here's your SRP receptor, right? Binds right there. And then you've got this translocon. And the polypeptide goes through the translocon, and then you've got the signal peptidase hanging out right here on the inside of the ER, which is going to cleave off the signal sequence, and then you end up with your mature protein in the ER. So that's pretty much how it all happens. Any questions about this? Okay. So the ribosome brings the whole complex to the ER by binding to the SRP receptor. So whenever something's going somewhere, there's usually something there's usually a protein it binds or something like that. She always there's not just random diffusion through the cell. Things are pretty directed. So SRP binds to SRP receptor. Okay, so you can think of it as there are two ribosomes, right? There's the free cytosolic ribosomes, and what kind of proteins are they translating? Are they translating anything? Anyone? I hear lots of people saying things, but
They're translating proteins without the signal sequence.
They're translating proteins without signal sequences that target to the ER. That's one thing. But where would those proteins be going? Do they just hang out in the cytoplasm? You remember that first slide I showed you, and it was like proteins can go to the nucleus, to the mitochondria. Like, remember, there's a whole cell out there. Whole cell. Proteins that function all over the place. So these free ribosomes are translating proteins that have other places to go. And don't forget, you have that post-translational import. So sometimes they're also translating proteins that are going to make their way to the ER anyways. So I want you to appreciate that ribosomes do not just sit on the ER and translate only into the ER. That's only a certain pathway called the secretory pathway. Okay? Okay, so here's your free ribosome cycle, which is exactly what you think it is, right? Ribosomes bind mRNA, translate proteins. And then there's the SRP cycle where they bind to the ER signal sequence and bring everything and translate into the ER. Two different pathways. So the ribosome binds to a translocon and translates the protein through the translocon into the ER, right? Protein, can it go just into the ER through the ER membrane? Can it pass right through? No. So it needs to go through some kind of channel. Yes?
So the ribosomes that are bound to the ER aren't permanently bound there, they're just kind of dragged there.
Mm-hmm. Yep. Thank you. Right. So it's easy to think rough ER has ribosomes bound, and that's just how it is, right? No, actually, it's just that was a snapshot, right? So those ribosomes are coming and going, because once they're done doing what they need to do, They leave and go find something else to translate. Great question. Okay, so here's a nice figure for those reading the paper. Who's decided they didn't want to read the paper for recitation? Okay, just making sure. So this is kind of nice because it's comparing co-translational and post-translational translocation. Because there were a lot of questions on Monday that were like, wait a minute, is the SRP involved with post-translational? And I was like, no. But everyone's like, well, how does it get there? So there's a nice figure in the textbook and a little blurb about that. But like, I really love the co-translational. So if you had to focus on one kind of translocation, co-translational would be the one to focus on in terms of my exams. But that's a nice comparison there. Okay, so I said that the signal sequence is removed. That's done by a signal peptidase, right? Signal peptidase. You can't go wrong with that, right? Because ACE enzyme, signal peptidase removes the signal sequence, right? Okay, and this happens in the ER, and it releases the protein in the ER, right? So you'll have your signal sequence here. And here, obviously, your SRP and your ribosome are not in this picture because it's just showing the detail of this part of it. But you'll have your signal sequence will bind to your translocon, and then the rest of your protein will be translated in. So you see how this loop is getting longer. And then the signal peptidase is hanging out right there, and it cleaves it off, which lets that fall down. And then once translation is done, you've got a free protein in the ER. Any questions about this process? Okay. Okay, so let's talk about membrane-spanning proteins, because I told you proteins that go through the whole secretory pathway are secreted proteins. Is the secreted protein going to have a membrane-spanning domain? Uh-oh, the lack of answers in this makes me really scared. Stop and think for a minute. Is a secreted protein going to have a transmembrane domain? No, thank you. Because if you have a transmembrane domain, where will you be? In the membrane. Thank you, guys. Okay. Whew. So you'll have secreted proteins, transmembrane proteins, and you'll have proteins that are going to the lysosome. Those are the proteins that go in the secretory pathway. So we need to talk about integral membrane spanning proteins, because how do they get inserted into the membrane? Right there during translation into the ER. That's where it happens. That ER membrane is the plasma membrane. Do you guys believe that? Do you think I'm lying to you? Are you going to go out there and say, Dr. Ehrlich is such a liar? She told us the ER membrane is the plasma membrane. Who believes me? Okay, good. Just making sure. Okay, so to have a transmembrane domain, basically you need about 20 to 30 amino acids with nonpolar side chains, and they're usually in an alpha helix where the H bonds are buried inside the helix, right? Because you need to be hydrophobic, so you're going to bury your anything that will H bond. So here's a hydropathy index. We saw this the other day. We see it again. And this is how you read it, right? So the more positive it is, the more hydrophobic it is. The more negative it is, the more hydrophilic it is. So this one right here, how many transmembrane domains do you think this protein has? Who thinks 1? Raise your hand. Who thinks 2? Who thinks none? Oh my God, there's only about 5 people in this class. Nobody wanted to commit. Okay, so when I say 20 to 30 amino acids that are highly hydrophobic, so this is your amino acid numbers. So if you draw that down to like 50, 60, 70, 70 something to like 100, that's about 30 amino acids going from here to here, highly hydrophobic. So that's 30 amino acids that are really hydrophobic. Maybe transmembrane domain? Who doesn't believe? Who wants to fight me? Okay. Well, yes. The answer is yes. Yes to what? I don't know. Okay. So I kept talking about topology and how microsomes tend to have the same topology as the ER, and how proteins have a certain topology, and how the topology is maintained all the way from the ER to the plasma membrane. And you guys are probably like, topology. What's topology mean? Huh?
Surface structure.
Surface structure. Yeah, surface structure. So let's talk about, when we're talking about proteins in a membrane, what does it mean? Well, if you think of what's in contact with the cytosol will always be in contact with the cytosol, then if the carboxy terminus of a protein is in contact with the cytosol, that is the end of the protein that will be in contact with the cytosol at the ER and at the plasma membrane. Okay? And we'll go through that and we'll draw it and we'll love it. Okay? So this is a different kinds of topology you can have. You can have your carboxy terminus in the cytosol, your amino terminus in the cytosol. You can have a really long cytosol. This would be your cytoplasmic tail, right? This would be a really short cytoplasmic tail. You can have these multi-pass transmembrane proteins, right? Lots of different things. But basically, I want you to think about it as what end of the protein is in contact with the cytosol, okay? And I'll use cytosol and cytoplasmic interchangeably, and I'll also use cell membrane and plasma membrane interchangeably, so just get used to it. Any questions about topology and why I'm talking about it at all? Has anyone, yeah?
What does topology mean?
So topology just means what's on the outside and what's on the inside. So if you're looking at the surface of the ER, here you would say the carboxy terminus is on the cytoplasmic side of the ER, and the amino terminus is on the luminal side of the ER. ER lumen means the inside of the ER. And this carboxy terminus is going to be on the cytoplasmic side even when it gets to the plasma membrane. So I'm going to harp on this, maintaining the same topology from the ER to the plasma membrane. Yep. Any other questions? Okay. Okay, so now we're going to go through this whole thing about how we integrate membrane proteins into the ER membrane and this and that. And so again, why are we talking about integrating membranes into the ER, integrating proteins into the ER membrane? Why would we do such a thing? Yeah.
Because when it goes to the plasma membrane, you may need transmembrane protein.
So if it's a transmembrane protein that lives on the plasma membrane, it needs to be in the membrane. So it happens co-translationally, right? So can anyone give me an example of a membrane protein that they can think of? Any kind of membrane protein. And it can be general or specific. Yes?
ATP synthase.
ATP synthase. Oh, bacterial membrane. Yes. Oh, no. Took me out of the eukaryotic cell. Yes, absolutely. Yes? Ion channels. Yep. Huh? A receptor. That's a great example. Yep, a nice general example. It didn't require you to know a protein name, but you covered a whole class of proteins. Absolutely. So these are things that need to be on the plasma membrane, right? A B cell receptor, a T cell receptor, an antibody, a toll-like receptor. Ion channels, which are usually made of multiple proteins in the plasma membrane. GLUT4, which brings glucose in from outside the cell, needs to be embedded in the membrane, right? Okay, so now that we have context, so there's a few things I want to point out when looking at these. One is always pay attention to carboxy terminus and amino terminus and what's where, because it's going to flip-flop, and that's important to understand because not all proteins are going to have the amino, just because the amino terminus is made first, and that might be close to where signal sequence is, doesn't mean the amino terminus is going to be on the on the inside of the ER. Oh my god, how am I going to make it through the next few hours? My throat hurts. Anyways. Okay. So it all starts with a start transfer sequence, which you can also think of as a signal sequence in this case, right? And it's going to be brought to the translocon, and it's actually going to bind to a part of the translocon. And then you're going to have, so you can imagine up here would be where your ribosome is. And so your ribosome is still translating. It's just showing you the end. And so it's going to bind here. Ribosome is going to keep translating, so it's going to push down, push down, push down, until you hit a stop transfer sequence. This is the yellow one. And that's going to bind to another part of the translocon. And your signal peptidase is going to cleave your signal sequence off, and that's going to fall out. And then the whole thing's going to move over into the membrane, and you'll end up with a membrane protein with a transmembrane domain spanning the ER membrane. In this case, the amino terminus is on the lumen, and the carboxy terminus is on the cytosolic side. So which side is going to be the cytoplasmic tail? Amino or carboxy? Okay, good. Yes?
So, when does the start transfer stop sequence? So it's right before, so it's on the ribosome, but right before it gets to the ER, that's when that happens, it transfers. I'm just trying to understand, like, I guess the before part, before it's...
So just imagine that you've got your ribosome up here translating, and you've got your signal sequence here, and the SRP grabs it, drags the whole thing over, and it starts to go in. But since it's a start transfer sequence also, it's going to bind right over here. Well, it binds to the translocon, and it stays there. And then the ribosome keeps translating, and as it translates, you can imagine this loop here getting longer, like you see right here. And then once you get to the stop transfer, then it moves over into the membrane. And then also the signal sequence is cleaved by the signal peptidase. Did that clarify? Okay. Questions? Okay. Okay, another way to think about it. I have a lot of figures about this because I think it can be kind of confusing. Does anyone agree with me that it can be kind of confusing? Okay, is anyone still wondering why you're learning it? Okay, well that's good. Okay, so this is just another way to look at it. So you've got, because this one has the ribosome in it, so I find that to be helpful. So you've got your mRNA and your ribosomes on it. Actually, it looks like polysome, right? Lots of ribosomes. And so your, so this is a little bit different, but anyways. So you'll have your signal sequence, which will bring it over here, and it gets cleaved off. So you can see it's already been cleaved off. So it's a different protein, so it's slightly different. But then you get your stop transfer sequence, which some people also call an anchor sequence. That might be more helpful. It's stop transfer as in it stops and it gets embedded in the membrane. Anchor, you can think of it as anchoring in the membrane. However it works for you, the terminology is different textbook to textbook. And then you, so it's cleaved off, you hit your stop transfer sequence, and then it's moved over. And you notice here your ribosome keeps actually translating, but it's going up, right, because it's kind of stuck right there. It's not going to push into the ER anymore. Okay? That's another way to look at it. So the stop transfer sequence, in this case, is defining the membrane spanning alpha helix. Okay, so you can have an internal signal sequence, which could be used to integrate a single pass transmembrane protein into the ER in two different ways. And so here you can see, here the amino terminus is on the cytosolic side, and here the carboxy terminus is on the cytosolic side. And what determines which way it's going to be flipped has to do with the amino acids surrounding the stop transfer sequence, or stop transfer sequence, is that what it's called here? So we'll go in a little bit more detail. So here you can see your, this is an internal signal sequence that's also acting as a stop transfer sequence, and right, so you don't see anything being cleaved here. Right, so it just binds right here to the translocon, and then the rest of it gets pushed through, and you end up with a transmembrane protein with your amino terminus on the cytosolic side and your carboxy terminus in the ER lumen. And the other way to think about it is with the C-terminal cytoplasmic tail, it's just the other one gets pushed in. The other side gets pushed in. So you start off like this, and then your internal signal sequence / stop transfer sequence binds to the translocon, and the amino side kind of gets pushed through, and your carboxy terminus is on the outside. Yes?
Is start transfer sequence kind of like a signal sequence in the sense that like it recognizes it and then finds there after it's already transferred?
The start transfer, so the start transfer sequence frequently is a signal sequence. Yes, it's recognized, it's involved in targeting, it binds to the translocon.
In this particular example, it looks like it's already synthesized.
In this case, it is, because it's internal.
Okay.
So when it's N-terminal, it's like just hanging out, SRP comes, drags it over. But when it's internal, there's a whole lot already made before it's out of the ribosome to be bound by SRP. And so SRP is not in this, but it is part of this process of bringing, whenever you think of binding to signal sequence and bringing it to the ER, always think SRP. Okay. Okay, so why would you care about N-terminal or C-terminal in the cytosol, or why does it matter? Why doesn't it all just go the same way?
Probably because we have like different type of transport. For example, co-transport.
Because there's different kinds of transport to the ER, is that what you're saying? Let's try another one. Thank you for contributing. Yes.
Does it have to do with charge?
Nope. No, so I think you guys are missing my question. I'm going to reword it. From the answers I'm getting, I'm thinking I'm being vague. Why sometimes you have the amino terminus cytoplasmic and sometimes you have the carboxy terminus cytoplasmic? Yes.
Is it because they bind to different things and they need a different size of
Okay, they bind to different things, so they're needed on different sides. So let's get more specific. What kind of things might an amino cytoplasmic domain bind to? So what kind of proteins would a cytoplasmic tail of a membrane protein bind to? Now, we have not done signaling yet, but there's a hint. Okay, so proteins involved in signaling. Whenever you have a receptor, why do you have a receptor? Just for fun? No, you're communicating a signal from the outside of the cell to the nucleus, right? So you have something binds to a receptor, and then you've got a whole bunch of magic that happens inside the cell that basically relays the message to the nucleus. And the first thing that happens are proteins bind to the cytoplasmic tail, right? I'm sure you've learned about this in very basic form before, but you've got these tails that dimerize and phosphorylate each other, and kinases come and phosphorylate. Yes? Semi familiar sounding a little bit? I don't know. Anyways, so what kind of things might bind to a extracellular side of a receptor? Give me an example. Think of a receptor and what might bind to it. Yeah. Like an antigen for an antibody on a B cell receptor, exactly. So the whole point of going through this is for you to understand that every protein has its own function and so a protein is not just a protein. Like each protein has different parts to it that interact with different things and do different things. They have different domains. So an cytoplasmic tail is going to need to be on the inside of a cell in the cytoplasm so it can function to relay a signal to the nucleus. An extracellular domain of a receptor is going to need to be on the outside so it can get the signal from outside the cell, right? If it's wrong, right? You do some kind of manipulation and you flip it around. Is that receptor going to work? Right? If you have an antibody where the signaling side is outside the cell and the antigen binding site is inside the cell, is that antibody going to do anything for the immune response? No. 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 Okay, I feel like I've lost all of you. I can't move on. Okay, someone tell me where they're confused. Either that or it's so simple, you're like, why is she going on and on? Is it that it's so simple?
I feel like you were asking us, why is it sometimes the carboxyside is inside and why is it sometimes the
Yes.
carboxyside is outside?
Yes.
But I don't feel like, as of yet, you've told us why.
Oh.
You've told us that
Oh, okay, good.
things are on the inside, things are on the outside, but you haven't told us why. That
Okay.
one is specifically on the inside sometimes and specifically on the outside.
Thank you. Thank you. Okay. I appreciate that. So, sometimes the carboxy terminus will be the part of the protein that, for example, let's stick with the B cell receptor antibody, because remember, okay, so you've got your, now mind you, right, so that's what your antibody looks like, and here would be your plasma membrane. Remind you there's different chains involved, but whatever. So, let's say, well, you can feel free to look up the sequence and find out what's the amino terminus and the carboxy terminus. But for the sake of explanation, let's say the amino terminus is here, right? So in this case, the amino terminus needs to be on the outside, right? Let's say, for some other, let's make a T cell receptor. Don't ask me how to draw that, right? T cell and B cell receptor are different. Let's say the carboxy, right, is that side. Right? So you need things to be oriented properly. So just because the amino terminus of a protein comes off the ribosome first doesn't mean that's the top or bottom or active site or whatever. Yes?
So you're saying depending on what type of protein they're going to be receiving, just dictates the properties of what the receptor will be?
Based on the function of the receptor, it dictates what part of the protein needs to be on what side of the membrane. No, let me change that. Based on the domains of the receptor. So we can say this is your antigen binding domain, and this is your cytoplasmic domain. Based on the fact that that's there and that's there, that dictates that this side needs to be out. Whether it's carboxy or amino terminus, who the hell cares? Because when it's all said and done, it's folded into some functional shape that binds to antigen. I heard someone say, oh. Does that mean someone got it?
Yeah.
Okay, are we good? Is that better? Did I answer?
Yeah, the way you first phrased it, it seemed like it mattered.
Okay.
Which side was in and which side was out.
It doesn't.
And
Right.
It doesn't.
Okay. It doesn't, but it's protein dependent. So it does for
does
each
per
individual
protein, but-
protein. Yes.
Can you say the O sentence again?
The O sentence.
Whatever you said to make him say oh.
Oh God, I have no idea. Because you're still confused? Okay.
I'm still on the page that carboxyl versus amino side matters, where you said it's actually.
It's protein specific. So what matters is protein's made, protein folds into a shape that's functional, right? It folds into its antibody shape. Who the hell cares what's on the amino side and what's on the carboxy side? What matters is that the side that binds the antigen is on the outside of the cell, right?
Yeah.
So it does matter. It does matter when it's going into the ER membrane, right? Because that dictates what's going to be on the inside and the outside once it gets to the plasma membrane. But the reason it's not always the amino is on the outside and the carboxy is on the inside or whatever, that's just dependent on the protein and where the action area of that protein is for whatever its function is. Okay, we got an O, we got a nodding yes. I see some people nodding. Are we getting it more? How about you? A little bit? Okay. You?
So if I want to recap what you said, pretty much saying that it doesn't matter which side is the carboxylate on.
It's not so much that it's dependent on which side is carboxy and which side is amino. It's dependent upon the fact that there's the part of the protein that needs to function in the place where it is. So the part of the protein that needs to be on the outside of the cell, like to bind antigen in the case of an antibody, is going to be the luminal side. Whether or not that's amino or carboxyl side of the protein, who the hell cares? What really matters is how it is when it's all folded and functions. But it's very important that it gets inserted into the ER membrane properly, because if it's flipped in the wrong way, you could end up with a cell like this. Okay, and you've got your bacteria up here and no antibody to bind it because the antibody ended up inside the cell by mistake. Okay? Yeah. Okay, come see me during office hours if you have questions. You can go.
Videos.